I try to imagine a fellow smarter than myself, and then I try to think, what would he do?
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. I am your host, Jesse Lawler, excited to bring you the 79th episode of this podcast, dedicated to the increasing improvement of your own brain by any and all means at your disposal. We're going to be talking about possibly the hardest to pronounce subject of any podcast episode that we've ever done on this show. Substance in question, I'm going to butcher this once again, is called Phosphatidylserine. Actually, that actually wasn't bad. That was pretty close to accurate. This substance is naturally occurring. It has both nootropic and generally physical beneficial properties to it. It's something that's present in the cell membrane of every cell in your body, so pretty baseline physiologically important, but topping up with it apparently has some pretty significant cognitive benefits, and that's what we're going to be talking about with Dr. Adam Parker, an expert on phosphatidylserine, whose name is mercifully easy to pronounce compared to the chemical that we're going to be talking about. That'll be in the main interview. If you hang around until the very end, in the ruthless listener retention gimmick, I'm going to tell you about an illicit stimulant that you probably haven't heard of, and how it's gotten interwoven with the civil war in Syria. So a little bit of stimulants in current events. Lots of other stuff as well. Interesting things going on in the world of Smart Drug Smarts recently. But in the meantime, as usual, let's start things off with This Week in Neuroscience.
Smart Drug Smarts, This Week in Neuroscience.
So I hemmed and hawed over whether to make this one an actual This Week in Neuroscience, because it's not like the case is closed. This is some particular new thing that we learned about the brain, but this just absolutely wowed me. And it actually is from this week. This just happened, I guess, very recently. But there was a teenager in Italy who dove off a bridge into a canal, got caught underwater, didn't pop up, his friends ran to get help. The kid was underwater for 42 minutes. Came up, was in a coma, of course, but actually did survive and is now up and running and neurologically functional again. 42 minutes without air, which just didn't seem real, but here it is, reported by the Milan Chronicle, which I assume is a major newspaper in the area of Milan, Italy. He was on life support for an entire month, but he woke up and seems to be doing fine. So this calls into question, obviously, how the heck this happened. How is it that 42 minutes without any fresh air, this kid survived, his brain survived, and it survived functionally? Scientists don't have a clear answer for this yet, but there's two things, each of which are probably involved to some extent. One is something called the diving reflex, or the bradycardic response, which is a physiological response that's been observed most strongly in aquatic mammals, but it's also believed to take place in humans. When such a mammal's face is submerged in water, blood vessels constrict and the heart slows down considerably. These changes sort of divert blood to the parts of the body that need the most, the internal organs. So you don't get as much blood to your limbs, the heart, the brain, the kidneys, these things get prioritized by the bradycardic response. This is apparently the same reflex that results in newborn babies holding their breath and opening their eyes if they're submerged in water. So that's one of the two things that could have saved this kid's life. The other thing is a selective brain cooling hypothesis, which essentially is looking at different people that have been through drowning or near drowning scenarios. And the belief is that the colder the water and the quicker the brain cools down, the more likely it is to survive for a long amount of time, said neurologist Dr. Zianka Falil. When you're immersed in cold water for a prolonged period of time, your body may carry out several processes that allow cooled blood to enter the brain. One of these processes, hypercapnic vasodilation, occurs when the body retains carbon dioxide as a result of not breathing. This extra carbon dioxide causes blood vessels in your brain to dilate, which in turn allows more cool blood to enter the brain. Needless to say, it's tough to exactly test this theory since not many people are volunteering to be near drowned, but the selective brain cooling hypothesis is considered to be a more likely explanation for how the brain might be protected during these episodes of prolonged submersion than the diving reflex. But either way, just as far as coffee table wow stuff, 42 minutes underwater, that's probably one of the two most, I just never would have guessed that statistics that I've come across since we started this podcast. The other one being the idea that the average person can live off no food for 60 to 70 days before they starve to death. Both of those I probably would have misjudged by at least like half an order of magnitude if somebody had asked me a couple of years ago. Had a banner week on iTunes, picking up some positive reviews from 3 different listeners, only 2 of whom I can even attempt to pronounce. One of them beat out Phosphatidylserine as having an absolutely unpronounceable username. Says listener Millie, if you're into nootropics and brain health, and by extension health in general, you will always have useful information after listening to this podcast. The app Axon makes it easy to keep up and is as well polished as the show. Thoroughly enlightening. Prayer Companion says a great source, a useful tool in the toolbox of life. And the absolutely unpronounceable account says, I frequent tons of sources about brain health and increasing cerebral function, and this podcast is consistently at the top of my list. So this kind of stuff makes me super, super happy. Thank you so much for each of you that took the time. Also, somebody that filled out the audience census said, I get more excited about new episodes of your podcast than the new episodes of the Game of Thrones. I didn't know that we were actually overtly competing with the Game of Thrones, but that brought a smile to our faces as well. By the way, thank you to everybody that did fill out the audience census last month. We got a bunch of responses for that, enough for some level of statistical significance. We're still tabulating all the results, but we'll have all those up and probably in some sort of visually pleasing format by the time next week's episode comes out and let you guys know what the demographics of our little community look like. So there is a ton of stuff going on right now in the Smart Drug Smarts universe that's worth talking about. I'm going to try to pound through some of the highlights here, not spend too much time on it so we can get on to the main interview, but wanted to let you know, if you remember a couple of months ago, we talked with Dr. Ken Ford, who is the founder of the Florida Institute for Human and Machine Cognition. Dr. Ford and his whole team just got a great write-up in the June 8th edition of Time magazine, which is about to come out, a cover story on the work that they're doing with humanoid robots. Dr. Ford was kind enough to send me an advanced copy of the article. I'm not sure if I can actually post this on the website without violating all sorts of Time copyrights, but I imagine Time magazine is easy enough to get a hold of, so just try to grab the June 8th edition and check out the article, which is really, really interesting. Also in June, the 18th to the 20th, I am going to be in San Francisco for the Quantified Self Conference, which I'm super excited about. We're going to be certainly doing some interviews and coverage of that event, and there will be one or two really cool announcements that are going to come out of that, so stay tuned for that in a not terribly distant Smart Drug Smarts episode. They are still selling tickets for that, so if you're in the Bay Area or you want to fly to the Bay Area and check it out, if you wind up making it out there, definitely drop me a ping and let me know. I would love to meet any Smart Drug Smarts listeners that make it to that event, and I'll be around all 3 days, the 18th through the 20th. And finally, a little homegrown event coming up this very Monday. So Monday, June 8th, we're doing something which I think we'll start doing on a fairly frequent basis. I'm really a big fan of Twitter, and we're going to be doing our first Twitter chat with Smart Drug Smarts alumnus, Dr. Patricia Gerbarg, who you might remember from the Rhodiola rosea episode. And we're going to be doing a big Q&A session at 8 p.m. Eastern Standard Time, Monday the 8th of June, all about Rhodiola rosea. So this is something that I've been taking on occasion ever since we did that episode. We've gotten a lot of emails and things like that wondering about some follow-on information about that substance, and so we've asked Dr. Gerbarg to be our first Twitter chat special guest. So we're going to be doing this under the hashtag AxonChat, and if you go to smartdrugsmarts.com/AxonChat, there's a little primer there. It's really, really simple, but if you've never done a Twitter chat before and you kind of want to know how the whole thing works, smartdrugsmarts.com/AxonChat explains what's going on, and basically you just show up on Twitter at that time, subscribe to the hashtag feed for AxonChat, and that's kind of all there is to it. And that's the first of what I expect will be many Twitter chats to come. So there's the tip of the iceberg for what we got going on right now. Let's move on to the main interview. Wait, hold on, I lied. One more thing. In the next couple of days, I'm thinking this weekend, I'm thinking probably like the 7th of June, we're going to be releasing the first ever Smart Drug Smarts Overdose Edition. I talked a little bit about this. We're going to be releasing this only through the Axon mobile app. So if you haven't yet downloaded Axon, if you got an iPhone, if you got an iPad, grab our mobile app. We're going to be starting to put out a little bit of bonus content through that app, and our inaugural Overdose Edition is just about to come out. So I will say no more. I will leave it at that. But if you're one of those people that prefer Smart Drug Smarts to Game of Thrones, then definitely get yourself to the iTunes store and download, and we will have a little something extra for you.
Smart Drug Smarts.
Okay, so we're going to be talking with Dr. Adam Parker from Angelo State University in Texas. He's an assistant professor in the Department of Kinesiology and teaches at the Ben Kelly Center for Human Performance, where he's also the faculty advisor for RAM Powerlifting and the Climbing Club, among other things. So he's really into physical fitness, and in fact, the study that he did into our star chemical for this episode, phosphatidylserine, involved its effects on both cognition and mood and how those interplay with physical exercise. But phosphatidylserine, as I mentioned, is a naturally occurring compound that's actually present in all living cells, both animal, vegetable, bacteria, everything. But it's been found to have a lot of effect actually as a supplement. And it used to be that when people were taking phosphatidylserine supplements, they were getting it from cow brains, which had pretty high squeeze factor. That's not the case anymore, as we'll hear in the interview. So let's go ahead and dive in.
Phosphatidylserine is a phospholipid that's integrated into the cell membrane itself. It helps with cell signaling, so when cells talk to each other or to themselves, phosphatidylserine is involved in some of those processes. It also is involved in prevention of apoptosis, which is programmed cell death, especially in neurons. We don't want them to die all that often. Sometimes cells need to die when they age enough. So the PS is involved in preventing apoptosis. There's some research that shows that it may be involved in helping to integrate undifferentiated satellite cells or the stem cells that could become neurons later on. So there's quite a few different ways that it can potentially work. The main thing, though, is in the cell signaling pathways and allowing those neurons to function effectively.
I read something interesting about that, how it's normally on the inside of the cell membrane when a cell is alive, but basically they can sort of flip around and be visible from the outside of a cell after a cell dies, and that's one of the ways that macrophages within the body know that, hey, this cell can be sort of eaten and recycled for spare parts.
Right. That's kind of what some of the research is showing. That's true. It's integrated in cell membranes of all living things. So eating anything, essentially, you're going to be consuming some amount of phosphatidylserine. But it seems that a lot more of it's going to be available in organ meats. It used to be derived from cow brains. So it was always called the bovine-derived phosphatidylserine. And in the 80s and 90s, with the whole issue with mad cow disease, or the real term for it is bovine spongiform encephalitis, they stopped producing phosphatidylserine from cow brain, and now it's derived from soy lecithin. So we get it from soy now. That does change a bit about the phospholipid itself. When it comes from animal products, it has different fatty acids on it compared to when it comes from soy. But it seems that our bodies can actually convert or change the fatty acids attached to the phosphate head, essentially, and make it into whatever it needs to be.
As far as its uses within the body, it sounded like something that was specifically very important for neurons. But organ meats, obviously, we don't think of organs as having a heck of a lot of neurons in them. What's the disconnect there?
Honestly, I didn't come across research and don't really know or paid much attention to why it's found in organ meats. I'm assuming just because the nature of the cells involved, perhaps. But honestly, I really couldn't answer that question.
Most of the food sources that I saw listed that contain it are meats rather than, you named soy, and that's sort of the standout that was not a meat on that list, but something that would generally not so prevalent in most fruits and vegetables.
Right. It's not found as commonly, but again, it's going to be found in any living organism. It seems that it's in higher concentration in meats, like you mentioned, but particularly in organ meats, kidneys, the brain, obviously. I think it's also found in higher concentrations in testicles or testes. Not that many people are eating those on a regular basis, but...
Right. So tell me a little about the studies that you've done.
The one study, I've only actually done one with phosphatidylserine, and I'm actually wrapping up another one with another supplement that's similar. It's called alpha-glycerol phosphorylcholine, which is more of a supplier of choline or phosphatidylcholine for the body. But anyway, back to the phosphatidylserine study. I did this, it's actually been over 5 years now. We took 18 college-aged males specifically because we were measuring testosterone. We didn't look at males and females. But we had these 18 guys take either a placebo or phosphatidylserine at 400 milligrams a day. They did both scenarios. So they either took the placebo first and then the phosphatidylserine second, or the phosphatidylserine first and the placebo second. But either way, they took the supplement or the placebo for 2 weeks at 400 milligrams a day. And then they came in to our lab for a testing session in which they started off with doing something called a serial subtraction test. So you get a random 4-digit number, and then you subtract the number 7 from that repeatedly for 2 minutes. The way I calculate this score is I count up the number of correct calculations and then divide that number into 120 seconds to get the average time per correct calculation. Then I also will score the total number of correct calculations and also the total number of incorrect calculations or mistakes. So if someone makes a mistake, we don't stop the test. We just let them continue from that incorrect number and see if their next number is correct. They also performed something called a profile of mood states questionnaire, which measures 6 different moods, and then it gives an overall score. So if I recall correctly, the moods are tension, anger, depression, vigor, fatigue, and confusion. And then there's a total mood disturbance, which is the overall score. So we had them do, again, the serial subtraction test, the profile of mood states questionnaire, and then they did a really exhaustive bout of lower body exercise. So they did 5 sets of 10 on 3 different leg exercises, and that included a squat and a Smith machine, leg press, and then leg extension. And the weights that they used were all 70% of their predetermined 1-rep max, so the most amount of weight they could do. And then they had a 2-minute break in between each set, and I think 3 minutes in between each exercise. So very short rest periods, very high volume, and kind of the reason why we used such an intense training protocol was to try to elicit a hormonal response. So when people exercise, it's a stress on the body, so cortisol levels increase quite a bit. But in males, much more so than females, testosterone levels increase quite a bit as well after exercise. So we wanted to see if phosphatidylserine had any effect on the hormonal response in these guys in response to that training bout. Then again, after exercise, they came back into the lab after working out. They did another serial subtraction test and another profile of mood states questionnaire after 5 minutes of recovery. and then again after an hour of recovery. And then throughout that hour that they're resting, we also collected blood samples to use to measure their testosterone and cortisol levels. And so the findings that we found were that when the subjects were on the phosphatidylserine, their field subtraction test score was significantly faster prior to exercise, but after exercise, that difference went away. Lots of research that shows that exercise improves cognitive function, and it seemed to totally negate the effects of phosphatidylserine, but before exercise, the phosphatidylserine led to a significantly improved brain function, essentially.
Yeah, I read a book, this was a couple years ago, called Spark, that was a survey of all the different research showing the various ways that exercise can really have all sorts of cognitive benefits, and it seemed like the higher intensity exercise, the benefits just got stronger.
Right. A lot of it is likely due to improved brain blood flow. The brain always gets a certain percentage of blood flow total, so during exercise there's an increase in cardiac output. And therefore, if the brain is still getting its 10 or 15%, or don't recall the exact percentage, but if it's getting that same percentage of a larger total blood volume, there's just going to be more oxygen availability and nutrient availability for the brain to function more effectively. I want to say the actual effect size, the actual difference in scores with phosphatidylserine versus the placebo was 21% improved time, and that was 1.35 seconds per calculation faster.
So do we have an understanding of what the mechanism of action is that leads to those benefits?
I don't think it's been really clearly established on how that works, but theoretically you'd think about improved neuron function, either through the cell signaling pathways. So cell signaling pathways oftentimes are involved with the adding of a phosphate group to something. So once that gets that phosphate added to it, it kind of activates it. So we talk, I guess the most familiar one might be ATP, our energy source. ADP is not a source of energy for us, but when we add a phosphate to it, now we can use that. That third phosphate can be used to be broken off and make energy. So when we phosphorylate or add a phosphate to something in the cell, that leads to these eventual activations of DNA being transcribed into messenger RNA, and then that mRNA being translated into actual protein. So if we can enhance the cell signaling pathways, you can improve the actual activity of the cell itself in regards to protein formation. So that's kind of where that comes into play. And the other big thing is just if we can keep those neurons alive longer, there's more of them available to function. And if they're enhancing the ability for those neurons to talk to other neurons, then that's going to improve our ability to make and store memories. I don't think there's any research that really shows for sure how these increased levels of phosphatidylserine in neurons makes memory improve. But there is a ton of research that shows it does, just not necessarily how it works.
Is there any sort of upper limit for safety or something that people should be aware of?
Phosphatidylserine does have FDA recognition as being generally recognized as safe. You can always look those up for any sort of supplement you're interested in. Just in a Google search for the supplement and then type in GRAS, and you can find anyone's submissions for that, and you can see if it was approved. So the approval for phosphatidylserine on the one generally recognized as safe document I came across said that it's approved for up to 300 milligrams a day. Likely there is some level that is dangerous in certain gram quantities you may be consuming, but at the 400 to 800 milligrams a day, most people would probably be taking, or maybe even as low as 200 milligrams per day. There's not likely to be a negative side effect from that.
And tell us about some of the people that are using this for more athletic performance than cognitive performance, because that's sort of what it's typically thought of as being related to.
Not necessarily just that. I think some people use it for the cognitive aspect and others use it for the athletic performance aspect. So there's been some studies that found that the phosphatidylserine helped to reduce the cortisol response to training. So anytime cortisol levels go up, cortisol is known as a catabolic hormone. So it actually causes the breakdown of muscle tissue. And most athletes obviously want to prevent that from occurring. So by blunting the cortisol response to training, there may be, in essence, a muscle protein sparing effect. So if we can prevent muscle protein breakdown and therefore increase the likelihood of having muscle protein synthesis occur, we're more likely to gain lean body mass. So some of the research shows that phosphatidylserine does in fact reduce cortisol levels. In my particular study, we didn't find that same finding. There was no significant differences in testosterone or cortisol after exercise or before exercise when the individuals in the study were on the phosphatidylserine versus placebo. Again, that may be due to differences. Some of the older research was done with the bovine-derived phosphatidylserine. Obviously, the doses could be different. There's several other issues involved with hormone levels. We have pretty large variations in hormone levels throughout the day. When you wake up in the morning, cortisol levels are really, really high in response to that stress of waking up, and then they kind of decline throughout the day. So Some research shows it's beneficial in regards to in reducing cortisol response, and other studies don't find that same result. I would think of it myself as more of a potential for cognitive improvement as opposed to athletic improvement.
What have studies shown on the effects for cognition for people that are older and maybe not in their mental prime of life?
Yeah, actually, the vast majority of research on phosphatidylserine is on older individuals, either with reported memory issues or that actually are suffering from Alzheimer's disease or dementia. And it seems that in the vast majority of research that I came across, and I'm talking somewhere between 15 and 20 articles, that phosphatidylserine either by itself or combined with other supplements, or a lot of the research is also looking at phosphatidylserine with DHA, one of the omega-3 fatty acids specifically. So when those are combined, the DHA is actually incorporated into the fatty acid tail of the phospholipid that is phosphatidylserine. So again, the vast majority of that research found significant improvements in memory or cognition or some other measure that they're coming across.
And phosphatidylserine was actually given sort of the stamp of approval by the FDA for people to put on the outside of packages that this can have cognitive benefits. And one step up from a lot of the supplements that the FDA is very cagey about allowing you to say anything overtly nice about.
With any supplement, you can't have the overt medical claims. Anytime you can see the FDA step behind a product and say that there is some research that shows this works, obviously that may be something that a person may be more likely to consider trying to see if it works for them.
So you mentioned that normally when people are supplementing, it's now with a soy-derived version of phosphatidylserine. What do we know about the potential differences as far as how that might be used by the body?
The way it's used by the body, it seems to be the same. The big difference is the actual fatty acid tails on it are going to be different forms of fatty acids than found in animal products. But it seems that our bodies can switch those fatty acids out as they need to be. Now, if I was going to be taking a phosphatidylserine supplement, I personally would probably try to go for the phosphatidylserine that's incorporated with the DHA. So there's a lot of the research that shows consuming either phosphatidylserine with the DHA or even just DHA alone being provided to cell cultures. So they took a certain strain of neuron cell in a petri dish essentially, and they provide that neuron with DHA, and the phosphatidylserine content of those neurons improved. So I would recommend then that you take a phosphatidylserine that has a DHA attached to it, or maybe perhaps take it with a fish oil supplement so that you have those available at the same time in the body. So it seems that the body will take the soy-derived phosphatidylserine just fine, and then if it needs to, it can switch out those fatty acid tails to whatever it needs to have available for the body's cells function.
Now, you mentioned in your study you had a loading time of about 2 weeks that the students were taking this before they did the measurement. Are there other studies that show how long the sort of onset period is before you can see effects, or do we know much about that?
No, I don't know that there's a lot of research that looks specifically at how long you need to be taking it for before it starts working. With the study that I just wrapped up with the alpha-glycerylphosphorylcholine, or AGPC, or alpha-GPC, we just use an acute dose, 200 milligram dose or a 400 milligram dose. We used 2 different doses of it and waited only 30 minutes. So that particular supplement gets into the bloodstream very quickly. Compared to phosphatidylserine, I'm not certain as far as the pharmacokinetics go, how quickly does it get into the blood, and then how quickly can it be incorporated into cells. Generally speaking, I think with a lot of this stuff, it might take some time for it to build up to beneficial levels. It wouldn't be something you just take right before an exam or right before some sort of stressful event. It might be something you'd want to have in your system for several days at least beforehand.
If you had a dream study that you could do next on phosphatidylserine, what would it be?
I think that with anything, you just try to improve what you've done before, what other people have done before. I think that there's been enough out there now that we know that phosphatidylserine seems to improve cognitive function. So I think it would be more useful to maybe look at a better dose, of what minimal dose is effective to have the benefits. That's always important to look at. Are there some other supplements that might improve phosphatidylserine's performance? For instance, with, adding the DHA to it, if it has to have it incorporated in the supplement itself, or if you could take fish oil along with it. That might have an additive effect or maybe even a synergistic effect with the phosphatidylserine.
So knowing what you know on phosphatidylserine, are you supplementing with it yourself nowadays?
Actually, no, I don't. Not that I wouldn't want to, it's just that it is a little bit of more of a pricey supplement. So typically, off the shelves, a supplement with phosphatidylserine is going to come somewhere between $30 and $60 a month. If I had, say, a parent or a friend that, suffering from dementia, I would definitely recommend them to take it. But I myself don't take that supplement. The only supplements I really take just kind of multivitamins, fish oil supplements. I take creatine because I'm into weightlifting quite a bit, and also supplement my diet with whey protein. As I age, that might be something I definitely consider. At this point in time, I don't feel like I've had too much of a cognitive decline yet, but I know that that's around the corner for everybody. So it is something I would recommend. Again, there's a ton of other supplements out there as far as improving brain health. the AGPC that I've been mentioning quite a bit now, that has some favorable research. Alpha GPC or AGPC, it serves as a phosphatidylcholine source, and then that choline then can be used to produce acetylcholine in the brain. But if you go back and look at phosphatidylserine as well, phosphatidylcholine is one of the body's sources for producing phosphatidylserine. So the choline is just replaced with the serine, and then now you have phosphatidylserine. So, in addition to improving acetylcholine production, which is one of the big neurotransmitters in the brain, and it's also the one responsible for muscle contractions and other parasympathetic nervous system function. So if you're improving acetylcholine production, but then also providing another source for phosphatidylserine production, it seems like that might be a potentially beneficial supplement as well. And in some countries, AGPC is actually a prescription medication. In the U.S., it's only found as a supplement.
It's so interesting how that works. Just completely flip-flops, and there doesn't seem to be much rhyme or reason to it, how some things are supplements in one country and prescription in another, and then you flip countries and totally different things are thought to be more dangerous or more regulated.
I teach a sports nutrition class. We talk a lot about supplements, but more in regards to improving sports performance. And in the U.S., steroids are a big no-no. But in other countries, we just drive across the border to Mexico and anybody can go to a pharmacy and order them. So, there's obviously negative side effects with anything, but steroids definitely seem to improve sports performance quite a bit. The big thing is we don't want kids taking them. Some countries, it's easy to get something and it's not seen as such as a bad drug. Other countries, it's seen as being something really bad and hard to get a hold of. It's kind of funny how it works.
Smart Drug Smarts.
So thank you so very much to Dr. Adam Parker for taking the time for that interview. One thing that struck me, I just re-listened to the interview actually, and Dr. Parker was fairly nonchalant about the results from his study, but actually it seems like a 21% improvement in the speed of cognitive processing on that sequential subtraction test. That's not nothing. I mean, that's quite a striking result if something is working 20% faster. We talked on the last episode about the use of the word significant in science versus in regular speech, and I would say that result qualifies for both uses of the term. It was a significant result scientifically and also a significant result as far as the actual effects. Also, some really interesting things that he had to say at the end there about alpha GPC, which is a compound that we haven't actually done an episode on yet, but I'd certainly like to. So if you're listening, if you are an alpha GPC expert, consider this an open invitation to introduce yourself, because I would love to do that compound also. And now, the ruthless listener retention gimmick.
Smart Drug Smarts. Ruthless listener retention gimmick.
So as if the Middle East did not have enough problems already, apparently there is a very, very popular, widely used amphetamine stimulant that is being used by all sides of the civil war in Syria. It's suspected to be being used to finance weapons sales, and it is certainly being used by combatants on all sides to be able to stay awake longer, fight with more ferocity, etc., etc. The substance is called phenethylene. It's a synthetic stimulant that was first produced in the 1960s as a treatment for hyperactivity, narcolepsy, and depression. But by the 1980s, it was banned in most parts of the world as being way too addictive. It's illegal in the Middle East, but is still hugely, hugely popular there. Saudi Arabia alone seizes about 55 million phenethylene tablets per year, and they estimate that that's only about 10% of the total amount smuggled into the kingdom. So a hugely popular thing. I mean, that's 500 million tablets. These things apparently go for about $20 a tablet street price. Lebanese psychiatrist Ramzi Haddad says that the effects of this drug are typical of those of a stimulant, producing a kind of euphoria. You're talkative, you don't sleep, you don't eat, and you're energetic. So if you've got some combatants that you want to fight, that might be a pretty attractive thing. Maybe no surprise that this is being used to the extent that it is. Interestingly, for those of us in the West that typically think that the worst problems in that region are caused by the super fundamentalist Islamic groups, it's actually those very groups that are least likely to be using phenethylene and other such stimulants because drug use is really frowned upon under Islamic law. So this amphetamine-fueled warfighting is actually more of a secular problem within that region.
Smart Drug Smarts. The podcast so smart, we have smart in our title. Twice.
Okay, so you heard it. That is the entire episode number 79. Thank you for hanging around until the end. If you liked what you heard, please tell a friend about Smart Drug Smarts. If you have two friends, tell two friends about Smart Drug Smarts. The show notes for this episode, as with all episodes, will be online at smartdrugsmarts.com, including links to everything we talked about here. Reminders are Quantified Self Conference in San Francisco, June 18th through the 20th. Twitter chat with Dr. Patricia Gerbarg on Monday, June 8th at 8 p.m. Eastern Standard Time. The inaugural Smart Drug Smarts Overdose Edition about to debut on the Axon mobile app. And Dr. Ken Ford getting a great write-up and cover story in Time Magazine on the June 8th edition. Hope you enjoyed this week's show. I will be back at you next week, as usual, with an unflagging, undiminished, and highly premeditated commitment to helping you fine-tune the performance of your own brain. Have a great week, and stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only. Although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.